Good morning, everyone, and welcome to yet another Pioneers in Biomedical Research seminar. Today, it's my real great pleasure to introduce Marcelo de Oliveira Dietrich, who has come to us from the Yale University School of Medicine, where he's an associate professor in the Department of Comparative Medicine. Marcelo's career and his interest really represent something that last night at dinner he referred to as integrative physiology. And it really also reflects something that I think neuroscientists often forget, but that is nevertheless really important. And that is that the brain is actually part of a fully complete organism rather than a disembodied entity outside of it. And accordingly, in his career, Marcelo has really done some remarkable pioneering work on looking at the relationship of neural circuits and neurons, taking advantage of the revitalization of the interest in neuronal diversity in the hypothalamus and other regions of the brain that control homeostatic behaviors that influence metabolism, obesity, exercise, many things that people here are also interested in. So I think his work really represents something that reflects the interfaces that we all think about in terms of how the brain influences organismal homeostasis and behavior. Marcelo is Brazilian originally. He did both his MD and his Ph.D. degree through a Brazilian university, although his PhD research was actually done at Yale in Tomas Horvath's lab, a pioneer in hypothalamic organization and circuitry. He's had a variety of experiences internationally, including a stint at the Cajal Institute, a stint in Austria. So he is a world traveler and ambassador of science. His awards include a NARSAD Young Investigator Award, several conference organization chairs, and also he is a Robert T. McCloskey Scholar at Yale School of Medicine. I think really the thing that really is remarkable about Marcelo's work is that he really has taken advantage of this idea that the brain is controlling something out there, not simply doing whatever it does. And I think the work accordingly, including some really exciting work recently on a little known region of the brain called the zona incerta and what it actually may be up to, reflects really the cutting edge of understanding how the brain controls the body that it lives in important ways. So Marcelo, thank you for coming from New Haven. And
thank you so much. Is this working? Yeah, everybody's here. Thank you so much. Yeah, thanks for the invitation. Thanks for the introduction. And I'm really having a great time since yesterday when I arrived. Not only in the town here, I walked this morning, but meeting people is so amazing, the science, and so amazing the resources and that you have here. Probably the students don't know that we never know what we have. We only know when we don't have. So, so just, I think there's a lot for you all to be proud and take advantage of what I have seen around here. So don't take that for granted because it's a privilege. And I think our job is a privilege that we can actually sit and be curious and go after what we are curious about. And when we see the reality of the world, I think we have to take responsibility of that because it's not everybody that has that privilege that we do. So I want to talk about, I started my lab at Yale about 10 years ago. And when I started my lab, I was very interested in development, in how neurocircuits regulate physiological and behavior processes. And I'm particularly interested in mammalian evolution. And one thing that always fascinated me is that one day I was a baby. One day I was a baby, I was being taken care by my mother, by my father, by my grandmother, and my neurons were doing something there to keep all the baby thing going on. And then I grew up, I became a teenager, and those same neurons were doing some other things. And then here I am with those baby neurons in my brain, and I always struggle, how is that happening? How are these neurons being able to solve all these different needs and problems that a living organism is having as we go through life? So that's really the question that I'm interested in my lab. I like to start with this video. So here you have the palm of a hand. It's a human. There's this structure here is an egg. This egg is the size of a penny, a 1 cent coin. So you are going to see here, what you're seeing is that there is this little animal hatching from the egg. It's a very immature animal. It's in a very primitive stage. This is not a bird. You can already see the hair. Only mammals have hair. So this is a mammal. And then you see here, there's already a white spot. That's the milk band. This little mammal already took its first meal, the colostrum from this mother. And here is the colostrum falling from the primitive mammary gland of the echidna. So echidna is one of the 2 species. There are many species of echidnas. This is the long nose. And then you have the platypus, the 2 mammals that are still on Earth today and lay eggs. So why I'm interested on that? Why I showed that video. As I said, there are 6,500 mammals on Earth today, species that are still extant. We have species under extinction every day. So the 6,500 is counting down, unfortunately. And we all come from a common ancestor that we don't know. We don't know what this common ancestor is, but we have many predictions of what would look like. It's probably some rodent type, small type, because it coexists with the dinosaurs. So was not eaten by the dinosaurs, would eat dinosaur probably eggs. So there was a coexistence there. And then it spread. Those are the monotremes, the egg laying, and then radiate to all the mammals. So what fascinates me is that despite all this diversity, different ecosystem, ecological niches that mammals, where mammals exist, different body size. We have large mammals, we have tiny shrews that are 2 grams, we have blue whales that are 100 meters long. All these mammals, as I said, the baby necessitates a caregiver to survive and to thrive. The extreme example is it's called a red-nosed seal that lives in the Arctic. These animals, they live in extreme weather, so they have the shortest period of lactation or mother-infant social relationship that we know. It's only 4 days. Those babies in 4 days, they need to drink half a ton of milk. They more than triple their body size. And then the mother will leave and the babies will go swim. And they will be weaned from the mother in 4 days. That's the shortest period that we know among all mammals. The interesting about those animals is that they are not ready. They cannot eat on their own just yet. But they cannot coexist with the mother because otherwise they are target of predation. So the mother leaves and they go. So they only swallow ice for another 30 days. And they burn all the fat until they develop the machinery and the capacity to eat. Why I'm saying that? I'm saying that despite the vast diversity of mammals, life on this planet has changed, the ecosystems have changed, but this feature in mammalian evolution that a baby necessitates a caregiver, even in extreme conditions, has not been wiped out of the mammalian evolution. So it's basically what one would say defines us as a group of animals. And the question I have is, how does the infant bond to their mother, to their caretaker. What are the neurobiological mechanisms for that? So I'm gonna give a little bit of a journey of how we think and the kind of key data that led us to think the way we are thinking in the lab. So I will cover a couple of stories here in this talk. So to start thinking about this problem, the most basically model that one can think is that you have a need, the baby has a need. For example, need for meal, for food. This need leads to a drive. That's the drive reduction theory from 1951. For example, hunger. And then the hunger leads to a behavior, for example, foraging, searching for food and eating, and that alleviates the need. So abstractly speaking, that could be a model. And as I said before, animals can eat on their own. They breastfeed. So you could basically substitute that model by milk, hunger, and then breastfeeding. So that was the simple model. It's a very Freudian model for the psychoanalysts or psychologists in the room. That was one of the ideas of Freud, that the milk and the breast is actually the primary resource of the caretaker, that the baby is attaching, creating all the derivative psychological processes. So many years ago, 10 years ago, we start testing this idea by studying these neurons, the HRP neurons in the hypothalamus. So this is the brain, the mouse brain. This is the base of the brain. Right here would be the pituitary gland, and then the sella turcica, bone, and then your oral cavity. So in this part of the brain, there are these neurons. This is the median eminence that connects to the pituitary gland. And these are the neurons that are more compact in this area of the brain. So these neurons were described in 1997. We discussed last night. So by Greg Barsh, actually, he was an Amgen. I checked. So he was Amgen in the pharmaceutical company when he described these neurons in 1997. Next to them, there's these other neurons, POMC neurons. HRP neurons are known now, and if you food deprive an animal, these neurons get activated, and that's important, leads to searching of food and eating behavior. So they are considered the bona fide hunger neurons in the brain. If you hear about a hunger neuron, the HRP neurons fit the model. And the POMC are the neurons that generate satiety, okay? So they are activated after a meal. For example, mutations in many of these pathways leads to severe human obesity. And Labradors, for example, dogs have a mutation in the POMC. So there's a 70% of the Labradors have a mutation, a truncated form of the POMC. So the satiety hormones are not produced. And the idea is that that's why Labradors are so trainable with food, because they don't have a satiety. So these pathways are highly conserved among mammals, and they play really substantial roles in human intake, human obesity, and other animals. So the idea was to test if what's happening in the babies is that when they are, for example, away from the caretaker, from their mother, they are hungry. So you are activating these hunger circuits, so they really want the breast milk back. And that's why you create this evolutionary bond. I will give a few key experiments that we named it for just be easy to talk. The first experiment is called separation, very simple. We take a baby mouse that's with the mother and the siblings, and we separate this baby for 2 different lengths of time to prevent milk ingestion. We did 8 hours. We thought that was quite a long time for a mouse pup to be without milk, and 90 minutes, which was a relatively short time for us. And we look for activation of this system with FOS, an early gene, counted the number of HRP neurons that were activated, and we were very surprised that these are 10 days old mice. They only obtain nutrition from the breast milk. That if you deprive a 10 days old, even as early as 90 minutes, you have profound activation of the hunger circuit. When you look and then you think, you say, okay, not big deal. Babies are always hungry anyway. So for a mouse pup to be away from 90 minutes without breast milk, that perhaps a very severe state of hunger. But you have to run controls. As we all know, controls are more important than the experimental groups in science. So the first control that I'm going to show is bottle feeding. The experiment of bottle feeding is basically we put a probe in the mouth of these pups, and then we pump milk to their mouth while they are separated. So we want to test if it's really the caloric deprivation, the lack of ingestion of breast milk that's leading to the activation of the system. So we label the milk with a dye, and then we find that they are actually ingesting. So the protocol works. They are actually ingesting this milk that's being pumped through this probe. And we do two types of milk. One milk, we milk the mother. So we pump milk from the mother and we infuse back to the baby. So it's the same mouse milk. And then we infuse we load the stomach with great amounts, so they are actually getting a lot of calories. And then we use also cow's milk, whole milk, as a second source of calories, as a different control. And that was a surprising experiment for us because we have here our nest, the animal is in the nest, very low activation of these neurons, no infusion, activate the hunger circuit again. But if you give calories back while the animal is alone, you don't prevent that. That kind of breaking our idea that, okay, it's the lack of calories that these animals are sensing, and then they are hungry, and then activating the hunger circuit. What we thought then, it was perhaps is happening is that when the animal is alone, the pup is predicting that because mom is not around, there will be no milk. So the pup is actually anticipating to being hungry. So it's working as a prediction. That's a different experiment to control, but here's what we try to do. So the experiment we call the grandmother. Okay, the grandmother we do like this. We take the cage and we take the mom away, and we put the grandmother. Basically a female that had her babies, and so it's a very good caretaker. But this grandmother doesn't have breast milk, so she doesn't give milk. So what we do, we basically feed the pups ourselves, based on the clock, like 24/7. We feed every 2 hours to 3 hours. We put a little rubber, so we make this fake nipple, and we teach them, they learn how to attach. We are not forcing the milk. We are not like, they learn to attach. They make the seal themselves, and you are going to see that the milk is going to change here. That is just the normal pressure and normal seal that the animal is doing to ingest the milk. So we feed them ourselves. And what's happening now? Every time we feed them, we put our hands in the cage, they come and climb to our hands to be fed. So they start developing an anticipatory response to us. We are the ones that feed them every 2 hours. And then we put them back in the cage, and the grandmother cares for them. So now we dissociate the social state from the feeding state. Being in the social world doesn't mean, is not related to being fed. Being alone with us means to be fed. And then we say, okay, if we take these pups and then we give a bolus of food, of milk, a very large bolus, and then we separate them, and some we put to the grandmother. If they are predicting the future caloric deficit, they all took the same meal, so they should have similar FOS. If there's something else that's happening in the grandmother cage that they are missing when they are alone, there will be a different activation. So something that's not related to the calories of the milk or the milk itself. And that was the case. When they are with the foster mother, the foster caretaker, they have lower FOS, and when they are isolated after this very large meal, they have this high activation. Suggesting that it's not the current or the future deficit of calories of breast milk that's activating this system, but the lack other stimulation that's happening in the social environment. So the question for us back in the day was, what are these neurons doing? What are these hunger neurons doing in early life? It's obvious that caretakers, mothers, give a lot more to the infants than breast milk. Stimulation is through that relationship that babies will learn about the world. And then I always like to revise the work of Harry Harlow when we talk about that, because Harry Harlow, despite doing some debatable experiments in the last part of his career that were ethically complicated ethically, he did really pioneer work in the early stage of his career in the 50s and 60s. And here was what was happening back in the day. So as I say, this Freudian theory of this attachment to the caretaker as a drive reduction from the breast milk was something that was not explaining child health and child disease. So if a child got sick in the 40s or 30s and will have to be hospitalized, mortality rates were 90 to 100%. So it was basically a death sentence. And the way the child care happened in hospitals back in the day was, this is pre-antibiotics, so it was like parents are not needed for the care of the babies. They need medicines, they need the food, and that's all. Then there was a doctor that heard about a hospital in Germany that had almost no mortality. So went to visit. He went to visit there. That's in the annals of the meeting of the American Pediatrics Association. So it's a meeting in New York in 1943 or 1944 that is that discussion. And they say, you visit this hospital and ask, what's happened here? What is the kind of treatment that you give that you have zero mortality rate? And the chief doctor said, nothing special. So, but tell me, well, when the child is very sick and we don't know what to do anymore, we have no treatment, we call Mrs. Anna. And say, who is Mrs. Anna? So no, Mrs. Anna is a retired woman. She volunteers and she cares for the babies. She holds them, she plays with them, and they recover. So that's when we start thinking as a scientific community that we have to change the way we approach the problem. And it's a very complicated problem because we try to be rational and give molecules and things like that, and doctors like treatments. And here is somebody that's saying that caring for a baby is actually the best treatment. So it's not providing milk to a baby through a bottle. So Harry Harlow tried to test those theories in the lab. So he was the pioneer researcher using rhesus monkeys. Nobody was using rhesus monkeys. Very few people were using rhesus monkeys back in the day. In fact, Yale was one of the first places using rhesus monkey. And there was a report by a woman scientist that she said it's interesting that baby orphan monkeys, when they are orphans, you cannot take their diapers. They fight when you try to remove their diapers because they get attached to the diapers. So the diaper, that piece of tissue, brings to this baby infant some comfort. So Harlow read that and say, wow, the babies are developing a relationship, a psychological relationship with a piece of cloth. Is that actually a mother substitute? So he wanted to test that in the lab. So he created two fake mothers. He took orphan monkeys, orphan rhesus monkeys, and then raised the monkeys in this cage with two mothers. I like to say that this is the Freudian mother, is a wired mother with a bottle of milk. And this is a cloth mother, is the same wired mother, but has a piece of cloth. Harlow was an interesting character, so he insisted that the mothers need to have a face. And then he asked the question, which mother would the baby monkey prefer? So when he quantified the the baby monkeys will spend 23 hours and a half a day with this mother and half an hour here getting the milk. So overwhelmingly showing that the preference for the baby monkeys for the clot mother. But that doesn't speak to us whether the clot mother has anything psychologically important for the baby monkey besides a piece of cloth that the baby is getting stimulation. So what I'm going to show you in the next 2 slides, 2 videos that to me summarize everything about Harlow's work and the importance of caretaking for the psychological development of the baby mammal. So Harlow realized that babies play in playgrounds. And if you have babies or familiar with the young kids, they play in the playgrounds and they are always checking on the caretaker who is around caring for them. If you disappear, they freak out. Okay, so they need that figure. So Harlow said, let's test that. So he made a playground and put the monkey alone in the playground with toys and the different mothers. So this first video is the Freudian mother, the mother with the bottle, the wired mother. You see that the infant monkey is frightened, is really scared, and will never play. He will stay in the crunch position and will never play. So he will take that monkey back to this cage, and now we'll release with the clot mother. The infant will directly go to the clot mother, and then we'll start playing. And oftentimes we'll look back to see that the attachment figure is there. So really, to me, that shows that it's not only that the baby monkey, the infant is spending time, is actually developing a psychological process in which that clot mother, in fact, works as an attachment, as a buffer for the distress of the infant. It's capable of buffering the distress and allowing the baby to relax and enjoy the world. So we wonder if the phenomenology that we were observing in the mouse is somewhat similar to this. So we use this work of Harlow as an inspiration. So the next experiment that we did, we call foster care. Foster care is like this. We separate the animals as we did for 90 minutes. And then in the other cage, we separate them and we put a different caretaker to see if these caretakers could suppress the activation of the nipple. We decided for 3 different types of caretaker. One was the grandmother type of things, a novel female that can care for the pups, but has no milk, no breast milk. In the mouse world, when the females are not lactating, the nipples are not protruded, so the pups cannot attach to the nipples. So we want to have a control for that, because as you know, there's the pacifier effect. Oral stimulation is very important. I like to show this because it's not only in the mouse world, it's in the human world, in the non-human primate world. Actually, many mammals, infant mammals, benefit from oral stimulation and the pacifier effect. If you are aware of working animal preservation, conservation, you know that in centers, for example, that take care of orphan bats, one way of making these bats to thrive when they are orphans is that you put a pacifier. They calm down and they survive. So we thought that a pacifier is also a phenomenology that we want to test. So what we did is to have some females that had protruded nipples, so the pups could attach to the nipple, but they could not take milk. There was no milk coming out. So they will have the oral stimulation. And the third group, we just swap to a different mother, lactating female. So just to control for this experimentation. And we look for activation of the hunger system, hunger neurons. Let's see. There's a little bit of a crash on my computer. I will start again. Interesting. I have a friend who went to the Brazilian army. We talked about the army last night. And he said his general told him, the best planning doesn't resist the first contact with the enemy. I always take that as a lesson. And I have presented this talk many times, and I don't know why this is collapsing. Let me do one thing. I'm so sorry about that. I will just remove all these animations from this slide just to try to show you. Otherwise, you have to trust me. Yeah. This is remove all the animation. So anyways, the nest has very little activation of the neurons. The isolated has high. If you give a lactating female, non-lactating female without nipples, you suppress. If the female has nipples, oral stimulation, you suppress even further activation. But if the female has or not milk, it makes no difference. So I show all these experiments to tell you that when that infant is alone, many things matter. The absence of many things matter, but the absence of breast milk does not seem to matter at all in many experiments that we have done, and is activating the neurons in the brain that are the hunger neurons in the adult brain. Yes?
Put the mom, the grandmom eat for the female. I'm thinking whether it's a safety issue, the baby feels safety. If you put the male, what happened? You tested that? Because I'm feeling the baby feel the safety issue. Yeah. When have the grandma or the safety issue causes that, you compare the milk and no milk. If the safety issue, it happened to the father or grandfather, what happened?
So let me show you some data, and then I will try to go back to that, okay? Because I think some of the data might help clarify, not fully, but to some extent. But I don't have the father data, but we have done. So I will let you, So everything that I show was early genes FOS, so we don't know the activity dynamics of the neurons. So we had the question, what is the activity dynamics in this process of being alone and being together, being alone and being together? So you could think about several models. We are only measuring the end point that the neurons are activated. It could be something slow, some thresholding mechanism. So to do that we did many, many years ago, was very challenging to set up these experiments to do photometry recordings, so recordings of neuronal activity with calcium in the arcuate nucleus. Is really the bottom of the brain. If there is a hard structure to get, is the arcuate. So when we had to do that in breastfeeding pups, like in 13, 14 days old. So this is the data, how it looks like. When we socially separate the pups, the activity of these neurons goes up immediately. And as soon as we reunite, the activity goes down. I'm going to show you a recording of this part of the data. So here is an isolated, this is a pre-weaning, is an 18 days old, 17 days old pup, a little older than we did the other experiments. The green signal is the real signal, the blue is a background signal as a control, and then we put here the mother, and you see this immediate response. The mother in this case is a new cage for her. She's not really caring for the pup, is just the presence of the mother in the environment. It's sufficient to suppress the activity. The puppy really goes after her, but she is not paying too much attention. So just the presence of the mother in the environment is important. So we did do several different social stimulus in this to see. So the mother is the most robust in suppressing the activity of the infants. Then siblings also do, and familiar females also do, and the father is somewhere here, is never as robust as the female. Okay, is never as robust as the mother. So it seems that there's something special about the mother. And that's just the statistics. I will cut a very long story short. So we test all different sensory components that are important for this reduction. And it's basically not much a surprise. It's driven by olfaction. So here we have a control pup and an anosmic pup that we made anosmic by metimazole. And then you are going to have the control in blue. We will introduce the mother, and then in this pink color, the anosmic animal recording of the HAP neurons. So you see, mother is around, and the control animals, the activity goes down, as in the previous slide, and in the anosmic animal, there's no effect. So the animal is making sense of the social world via the olfactory cues. At least that's the important information that reduce the activity of the HAP neuron.
Olfactory, or is it main olfactory system?
We don't know. Yeah, we don't know. Very good question. We really want to know that. There's a lot of other things that I will get later in the talk. And because I will get on the food. The smell of food affects these neurons, and the smell of social. So where these two informations coming from, and where do they it's a very important question, I think. So while we wonder there, you can find things, and that doesn't mean that they are important for anything. Like you do something to an animal, a neuron is activated, a neuron is inhibited, but that might not have any physiological relevance for anything. These are developing pups. Maybe this hunger circuit goes up and down, but doesn't actually have a fundamental importance for the pup itself. So as I say, what do babies do, right? So I like to go back. So around the same time as Harry Harlow was doing experimentation, there were two scientists that were really pioneers, human scientists, John Bowlby in the UK and Mary Ainsworth here in the United States. What did they pioneer? They pioneered the entire field of attachment behavior in human psychology. So, which Bowlby was inspired by all this animal work, and say that the most fundamental importance of a caretaker is to be a safety net. Is not the Freudian milk association, is to build a safety. The caretaker needs to be that the baby, the infant needs to develop an attachment figure where it finds safety for them to be able to develop normally. And Mary Ainsworth developed the tests that in the lab to test attachment styles or the degree that an infant is attached to the mother. So this video is one part of this test. I would try to, the audio is not working, so I will try to do this.
So caregivers in the
After
room.
a few minutes, the caregiver again leaves.
So caregivers in the room, baby's playing, caregiver's gonna leave the room, and the investigators are looking through a mirror, a glass window, what's gonna happen.
This time, the baby is alone.
So that's very upsetting. The baby's in distress, starts crying. Many baby mammals cry, not only the human babies. In the mouse world, mice also cry, but they cry in the ultrasonic range. We don't hear, but when they are alone or our babies are under distress, they vocalize in the ultrasonic range. So here is an example of a 12-days-old pup that's isolated. And these are the ultrasounds that we brought back to the oligo range.
This video is much slowed down. Sometimes you can see the mouth opening, and each of them is a cry, a syllable.
So we wonder if this HRP system is important for this distress response of the infants, for the emission, the infant cry, for the emission of ultrasounds. So we did as we do, usually in biology, we loss of function and we gain of function. So the first is loss of function, HRP neurons release GABA, we knock down the GABA transporter so they cannot release GABA, and we ask if the babies cry as much. And they vocalize much less. So if we knock down GABA released by HRP neurons and social isolate a baby, we find much less of this vocal behavior, suggesting that it's not only a byproduct of development or something. What we are finding is not just some random phenomenology. There is a physiological importance of the activation of the system. We also use chemogenetics and overactivate the neurons. So the pups are alone, and then we overactivate them, and then we can induce higher rate of cries. And then the question is this change of the behavior of the infant sufficient to create a change in the behavior of the caretaker? So the experiment that we did was, we have a mother in a treat chamber where we have two of the siblings. And we ask, does the mother prefer one of the siblings versus the other? And the answer is no. On average, it's 50-50. So we really don't find a preference when you do that test. But then what we do in a second stage is that we chemogenetically overactivate the neurons of one infant. So we drive that system high, high, high of the HRP neurons, as if the baby's playing being alone. And then we ask, does that change the behavior of the mother? So the mother will explore. We did this in the cups because mothers take the babies and bring back and care for them immediately. So we need to prevent that. So when we activate the neurons, these mothers go around this cup and they will bite the grid and they will really try to take the pup out of it. And they will spend most of their time here. So we can completely shift the preference between the two siblings to the one that we overactivate the neurons. Just suggesting that this, whatever is happening in the system in the pup brain is actually changing the physiology and the behavior response of the pup, and that's actually transmitted to the mother. Hence, it's very likely there's an adaptive value in the evolution of the circuit because it's changing the behavior of the caretaker. And that's just the quantification. So that's just the summary of this part. So we think that in the adults, what these hunger neurons do, the proximal causation, so what we are measuring in the lab is that these neurons sense food deprivation, motivate the animal to seek food and eat. Ultimately, that restores homeostasis, energy balance. That's the ultimate causation. That's what drove the evolution of the system. In the neonates, these neurons are, when they activated, they drive the motivation to seek proximity of the mother, not ingestion of milk itself. Every time the mother is around, there will be milk. So milk itself becomes an irrelevant stimulus in the evolution of these animals, but ultimately it's the same thing. It still restores homeostasis. Now, as I said before, development changes. The baby brain changes to an infant brain, to an adolescent brain, to an adult brain, and our neurons are in our head. The HRP neurons that are here today were here then. So we want to, so as I say, there's this early postnatal development, adolescence in the mouse. This period is particularly interesting because the social behaviors, these are rat play in the lab. The juvenile period, early adolescence, is a period with intense social behavior, intense play behaviors. Those behaviors don't have a clear homeostatic value, like the mother being around, there's a homeostatic value, but the animals playing, there's less so of a relationship with the homeostatic value. And then they turn into adults and then late adolescence. So the question that we had was, when do HRP neurons stop responding to this social isolation and reunion, since we know that that doesn't happen in the adult brain? So the experiment again, it's simple in that period, in the juvenile period when animals play a lot, they have this heightened sociability. We take some of these animals from a cage and we take the food out for 3 hours, 3 to 4 hours. So they are food deprived, but they are in social group. And we ask, is that sufficient to activate that food deprivation, activates the AGP neurons in juveniles? And we also run another group. Now we have the advantage that in this period of life, after weaning, they can eat. So you can socially isolate an animal without food depriving. We don't need to do the bottle feeding procedure. And we look for the activation of these neurons, the AGP neurons across age, 25, 35, 50, and after 65, 70, 75 days old. These are control animals that are in the home cage. Usually AGP neurons are not very active with FOS. This is if we food deprived for these 3 to 4 hours, we have a activation in the adults less so, but you still activate the system. And if you socially isolated the animals, you have in late adolescence, this very late adolescence, you still have some effect, but in adults, you don't have an effect. Even if you socially isolate the animals for a day or so, you don't activate these neurons ever with socially isolated. So there is a closure of this mechanism. That's just the statistics. And so the model that we have is that social isolation during this period of life leads to a social need and enhanced sociability, and that social need is encoded or necessitates the function of AGP neurons. To test that, what we did was this. We look for the sociability, for social interactions, the time the animals social interact after a period of social isolation. So we take juveniles that are very playful, we test their social investigation of a peer, and then we socially isolate them for 3 hours and we test them again. Usually, these periods of isolation are sufficient to increase sociability. And in another group of mice that express the inhibitory dread receptor in the AGP neurons, we inhibit the AGP neurons. So we prevent the activation of the AGP neurons during the period of social isolation and look for sociability. What I'm going to show here is the difference in the social investigation after social isolation and before. So the control group, the juveniles, they will socially investigate much more. They will interact much more after social isolation. When you inhibit the AGP neurons in juveniles, you completely block that effect of social isolation. So you can block the increase or the need, one would say, for the animal to socialize. When you do the same experiment in the adults, 75 days old animals, you still have an increase in sociability after the 3 hours of social isolation, but you have no effect of blockage of AGP neurons. To me, really showing that whatever we are seeing here is a very developmentally restricted phenomenology of how the mammalian or social brain is actually working to modulate social behaviors. We also perform a bunch of recordings, photometry recordings during juvenile period to test different social mates and different things. And so here I'm going to show reunion with a sibling, reunion with a toy, a rubber duck, and a food, and how that affects the activity of juveniles. So a social mate, a peer, inhibits the neurons similar to the mother in the pre-weaning animal. A toy doesn't do anything, and the food also inhibits. The effect of food and social are somewhat equivalent, and there is no correlation. So an animal might respond strongly for food, weakly for social. So suggesting at least that this might be independent processes, that the neurons are independently responsive to one or to the other. So I said the question is then, when do these hunger neurons stop responding to the social world? I show you some FOS data that in the adults, they are not activated by social isolation. So here we perform fiber photometry longitudinally through this entire period of development, reuniting the socially isolated animals with siblings in this case, and then you can find that around 50, 60 days old, you stop having response to the social environment, similar to the FOS data. They never respond to the toy, and the response to the food is fairly stable and will continue through adulthood. So I think I hope the data, the model that we have in our heads, that during this period of development from infancy to late adolescence, the AGIP neurons are encoding the social needs of animals. So how do you put that in a framework? What does it mean? What are we learning here? Does it, I think we always have to put things, at least for me, in a context of evolution. And I think the model that we try to work is with the model of exaptation. We try to understand the system as exaptation. For those that don't know what exaptation is, does not really fundamentally matter. I put this word here because I like to make a point. And the point that I like to make, this was a paper written by Stephen Good and Elizabeth Verba in 1982. And when they write the paper, it's a beautiful paper. Stephen Good was a great writer. And when he started writing the paper, he says, if you don't give a name to something, people don't see it. The phenomenon might be in front of you, but because it has no name, nobody observes. So they said, this is happening in evolution all the time. But in evolutionary biology, we are not talking about it because we have no name, so let's give a name. And the name is exaptation. So what is exaptation? And why do I like to make the point? It's also because when we talk about diversity, inclusion, and these concepts in science, is much more than bringing people from underserved backgrounds. Each of us with our different language, with our different upbringings, we see the world in a unique way. By bringing people together that see the world differently and investigating the same problem, people can see the world in a way that nobody else will be able to see it. That, to me, is the fundamental importance of bringing everybody together to solve problems that are important for human society. There's another, I will not talk about that, but I can talk later during lunch for the students about another example of an astronomer that went to the space station and how this concept is very important. Anyways, exaptation. Exaptation is the following phenomenon. Evolution does its job and evolves some features with some adaptive value. For example, you need some bones for mastication. So there was evolution of many bones for mastication. Somewhere along the lines, not all those bones were important for mastication anymore. So there was little pressure to evolve some of these bones. So this particular bone in purple start to have really reduced function in mastication as animals evolve. But somewhere along the lines, in the mammalian evolution, in two different processes, these bones were reutilized for a different purpose. They became our ear bones. It's how we hear. The three tiny little bones, if you have never seen that, is the most fascinating thing. Those three tiny little bones that make us hear. Those bones were originally masticatory bones. So what did the evolution do? It repurposed something that evolved originally for a reason to another, to fulfill another function. It repurposed, co-opted, exapted. Exaptation is the name given to that process. After this term was coined by them, exaptation, some people in evolutionary biology claim now that everything that we see is actually mostly exaptation. Even this bone, that things are just being repurposed all the time. So what we think is happening here is a phenomenon of developmental exaptation. In the evolution of mammals, mammals comes from ancestors, reptiles. This feeding circuit is there. A reptile that's hunger activates the AGP neuron. Now, you have to evolve a completely different feature features in the mammalian evolution. Now you have altricial, immature babies, they need a caretaker, they have this long development, the same neurons in the brain. You cannot come up with hundreds of different circuits. You fulfill the needs with what you are at hand. So you co-opt systems that were there for different purposes or different functions. So what we think is that these hunger circuits are being co-opted in mammalian evolution to serve a different function, a social function in early development, which is unique to mammals, this early developmental period. Hard question to try to answer, but we are trying to answer that by using comparative genomics, single cell genomics in reptiles and mammals, and to see if there is a particular evolution of a subtype of age-up neurons that respond to the social world. That closes the first. Do I still have a few minutes or should I stop?
3 minutes.
3 minutes.
So
maybe I go for the few extra slides. So that's all about the needs of the baby or the needs of the teenage. How is that social need being transmitted? But when that baby monkey goes to the playground, touches this cloth and calms down, that's a different story. It's how is the brain of that infant making sense of the sensory world to say, this is my attachment figure. I need to calm down. So I'm fascinated by that question. So I'm going to show some data briefly about this story on the zone inserter. So you can approach the problem in many ways, and we are approaching the problem in many ways, but what I'm going to tell is one way that we approach the problem. And one is like, the baby needs to be integrating sensory experience, the sensory world to make sense. This is an attachment figure. So we need to look at areas in the brain that integrate sensory experience. Now, the zone inserter, the name says that's zone of uncertainty, even though we know a lot about it now, is located in between the hypothalamus and the thalamus. It's this mustache-shaped structure. And this zone, there are two features that to me was very interesting. First, in the adult, they integrate a lot of sensory inputs and interoceptive. The second feature, neurons here, they highly innervate the neocortex, but during a very restricted period of early development. And then the projections retract. And if you kill those neurons in the mouse brain during early development, you affect pyramidal neuron development. So I thought those kind of an interesting features. It seems that there's a functional importance for these neurons. So we want to understand if the zone inserter was an area that's integrating the sensory experience to the social world. So we recorded 3 subsets of zone inserter neurons. Glutamatergic neurons, PV neurons, and somatostatin neurons with fiber photometry when the pups were interacting with the mother after a period of social isolation. As you can see, during the social interaction, these neurons are highly activated by the mother. It's contact that activates these neurons. So here's one trace. You can see the activity of the neurons. We put the mother, activates, and then every time there's a contact, there will be a peak. I will zoom here. This is the first contact. That's another contact. That's that activation. We try to control for that. Here's a free moving interaction. It's complicated to control. So what I'm going to show here is an experiment that we did. We put the pup in a track. The pup needs to run to the end of the track. And then the first try will see nothing. The second try will see a fluffy mouse toy, but it was just a toy. And in the third try will be the face of the anesthetized mother. So the mother was not moving around, but they would touch. And then the activity of the neurons will be from blue to yellow. Yellow is more activated, okay? In the first trial, the activity is kind of fluctuating, go to the end of the alley, nothing happens. We put this toy, goes there, not much. And then when you put the mother, as soon as it activates, touches the mother, the activity of these neurons goes really high. So they seem to be really related to this touching, the contact with the mother. These neurons respond to many other social stimulus not only the mother, the highest responses to the mother is even higher than to the father. Okay, so the response to the mother to the contact is always high. As I said before, pups cry. This is ultrasonic vocalization when they are isolated.
Is it physical contact or is it olfaction and proximity?
Is olfaction necessitates whisker and olfaction together.
Okay, so
If we whiskerectomize, we suppress very little. You can not even see. If you make an osmic, same. If you take both, gone. So it needs the two senses. Yeah. So as I say, baby's alone, sees the mother, touches the mother, activity of the neurons goes up. We want to test if that's a comforting, that really had the features of an attachment figure. So we activate the neurons, the zone inserted neurons when the babies are alone. Here is the baby alone crying. When we activate the neurons, basically shuts down vocal behavior. Actually, it's the strongest data we ever got in my lab studying this in a decade. They really stopped vocalization. We did other controls. It also suppresses corticosterone release. These animals are not releasing corticosterone. This is the isolated animals releasing stress hormone. When we activate the neurons, they are not induced, so it's suppressing that response. Here we did a loss of function. So these neurons, we express CUR 2.1 to these neurons. So in the control, they are with the alone, vocalizing. When they see the mother, we put the mother, they stop vocalizing. If we have a loss of function of these neurons, which means the mother cannot alter the activity of these neurons, you don't see an effect. So suggesting that the zone inserted neurons are, in fact, important to transmitting this information about the sensory world and buffering stress of the pup. And the most important data for me is learning. As I say, the infants learn about the world in the presence of a caretaker, of an attachment figure. That's the environment by which an infant can actually experience the world in a positive way. So we tried to check if the system was important for that. So the experiment we did, we conditioned the animals, the pups, with their anesthetized mother in the presence of a toy with a neutral odor. And then we did nothing, control animals, or inhibit the zone inserted neurons during this contact with the mother. If we don't do anything, the animals learn that the odor of that toy is a positive odor. So when we test next day for preference, they will prefer the odor of their parent. If we inhibit the zone inserted neurons during this experience, the social experience, they don't learn about the world. They don't learn that positive experience. So suggesting that whatever these neurons is doing is actually important for the cognitive development and learning of the infant. The circuit of these neurons is very wide. They receive inputs from many areas, send inputs to many areas. So we are interested in decoding that. Just one more slide. So one question that we have is like, how is the infant brain modeling the concept of a caretaker? Everything that we have done in our lab so far, which is actually a huge surprise because mice are generalists in terms of social behavior, but we always find an increased response in neuronal activity to the mother. Always, a little more than everything else. So there is an encoding of the mother. So the question is, how is the brain making that, encoding that information? To me, is a fascinating question. It's a complicated question to answer. So what we are trying to do is to record, not with calcium, but recording with silicon probes. So we do optotagging in the pup of these neurons, and then we can tag zone inserted neurons. So we know that's a zone inserted neuron, we know electrophysiologically. And then this is just an example of recording. As I said, these are contact neurons. Each spike sounds like a spike. Populations that are activated by the mother. So what we are trying to do right now is have enough data so we can build models of how the infant is actually encoding the information about the social world. That's how I want to end, where we are leading. This is my lab. We have a medium-sized lab. And actually, if anybody, students are interested on this, or if you know anybody, this is a great time. We are going through a little bit of a recycling. Grad students graduate, postdocs got jobs. So we will be looking for 1 or 2 new postdocs during this year. Thanks so much.
All right, since we're a little over time, anyone who needs to go, please feel free. We won't be taking names. And then we have some time for questions.
Are the HRP neurons also responsive to like hypoxia or sleep deprivation in adults, or is it just hunger?
Wow. Hypoxia, I don't know. The information about sleep is the one I'm trying to recall, but I cannot recall. But I think there's definitely a paper, a Nature Neuroscience paper that look for circadian oscillations, and I think they sleep deprived, but I don't remember. But it's a great question. I don't know.
Other questions?
Have you ever looking at the output mapping of the HRP neurons, because the neurons can responding to the food cue and also social cue. I just wonder whether there are some segregational population depending on the output target.
Yeah, we don't know yet. So we don't even know. These are different neurons that respond to food deprivation, to social isolation. So we are doing that in many different ways. It took us many years to actually be confident. Now we are doing trap. So with trap, with FOSS-TRAP, FOSS-CRE-R. Actually, FOSS-CRE-R with FLIPO. So we express FLIPO in the HRP neurons, all population. And then we express CRE only in the food activated or in the social isolated activated. And then we are doing single cell sequence and PET-seq and circuit input output behavior, but it's really ongoing. So I don't know. It seems that anatomically they are located in slightly different areas, which would suggest that everything is different from what we know about the HRP neurons. So, but it's really preliminary. Yeah.
Other questions? So I have a question. It's more of a speculative question, but for both the zona incerta and even the AGRP neurons, evolutionarily, clearly important adaptive and homeostatic behaviors have been assigned to a very small population of integrative neurons. And so I'm just wondering, whether you think that that's because you have to have such a safety factor that it has to be precisely mediated by a common population of neurons, or whether or not it's because you have to integrate so much information that you just have to have massive convergence on something. So in other words, is it the neuron identity of the AGRP or the zona inserted neurons that make possible these major homeostatic safe behaviors, or is it because they are the targets of massive input?
The way I think is the latter.
Mm-hmm.
I think it's I think the circuit organization
Mm-hmm.
of this population serves a purpose.
Mm-hmm.
And I think, yes, that's how I think. And I think there's a lot of ways that the brain can solve the problems. Mm-hmm. So if you affect these systems, the animal won't die.
Right.
It will develop other means. Mm-hmm. So, and I think in experimental research, we try to control everything. So we find the effect that tells the story that we want to tell, but there are a billion other ways to do the experiments that maybe we won't find that the AGP are so crucial, or the zona insert is so crucial. You understand what I mean?
Yeah, that's what I'm talking
that
about.
There's it's a much more dispersed network. Right. Now, for the zona insert, I think that's much more important because the zona insert is really integrating. The The thalamus is also integrating. But to extend that the zona inserter is competing with the developing thalamus in the early infancy, I think is an interesting idea that the zona inserter comes at play at a moment that the thalamus is still undergoing development for integration of the sense, for higher level integration. So you take the system, plays a role, and then the projections fall apart. So
Sort of like the subplate of the thalamus.
yeah, to some extent. And the HRP system, I think, is slightly different. If you think about hierarchical models or something like that, it seems to be higher in the hierarchy to generate this need state. They're not integrating anything. They are really generating a need state. Information is coming. When the information come, activate, you generate a need state that leads to a motivation, food, social, whatever. The way I think is that what these HRP neurons do is anything that relates to caloric counting, energy balance is transmitted to these neurons. So in the early life, socialization has a caloric value, you know, even for juveniles. Like there's a nice book by an anthropologist that investigated a certain tribe in Brazil of Yanomamis, and back before they were more mixed with our culture, like city-like culture or whatever, and they sleep together until the man sleep together. They hug each other because the nights are cold. So they sleep together and they hug each other until they're young adults, and then they stop it. When they are sexually mature and they will have partners, then they stop that. But until then, they use that as a form of actually warmth. And it's very interesting because the men that sleep together, they never fight later in life. They will fight with other men, but they will never fight with the man that they slept together. So to me, it's a very interesting that the social behaviors have this homeostatic value. That calorie is being computed.
That's the other question I was going to ask you is what's the adaptive purpose of the play social contact for this issue of, you know, metabolic homeostasis, et cetera. And so that kind of answer is warmth and expenditure of calories is minimized when you have fewer warmth.
Yeah, animals together. Yeah.
Any other questions? All right. Well, thank you, Marcelo. And